All right, so today we're going to do a deep dive into something that I think is super helpful for anyone who's studying GCSE biology.
Okay.
We are going to look at the June 2023 exam,
Great.
but more specifically, we're going to look at the parts where students tripped up.
Oh,
So we
excellent.
have the examiner's report, and it is fascinating.
I bet.
And trust me, it's way easier to learn from other people's mistakes than to make them yourself.
Yeah, absolutely. Like having a cheat sheet for how to not mess up the exam.
Exactly.
Love it.
Okay, so first up, we've got viruses and bacteria.
All right.
You'd think telling them apart would be simple, but question 1B showed that some students got them confused.
Yeah, it's easy to get those mixed up if you're not paying attention.
Yeah.
I mean, they're both microscopic.
Right, they're both a little bit,
Yeah.
foreign to us.
The big thing to remember here is the difference in their structure.
Okay.
Viruses are basically genetic material bundled up in a protein coat.
Okay.
The bacteria are a bit more complex. They've got a cell wall. They have cytoplasm, other structures. Okay.
Think of it this way. A virus is like a sneaky little package,
Okay.
and a bacteria is like a whole mini factory.
I love that.
Yeah.
That's a good analogy.
Hopefully that helps people remember. Be
Yeah.
really precise with your terms and your knowledge.
Yeah.
Especially in biology,
Yeah.
Knowing the specific features of different microorganisms is key.
Yeah. You don't want to be mixing up your fungi and your viruses.
No.
All right. So moving on to question 2,
Okay.
which focused on food webs and how populations change.
Okay.
Apparently, some students struggled with drawing food chains and showing the flow of energy correctly.
Oh, yeah. Those arrows can be tricky. I remember when I was learning this stuff,
Really?
I used to get it wrong all the time.
You got to remember, they always point from what's being eaten to what's doing the eating.
Okay.
You're following the energy as it moves up the food chain.
It's like a one-way street for energy.
Exactly, a one-way street.
There was this example in the report about what happens if the number of arthropods decreases.
Okay.
So fewer arthropods would mean less food for the birds that eat them.
Right. Right. So their population would likely go down.
Okay.
But the worms who are preyed on by arthropods would probably see their population increase because there's fewer predators around.
So it's all about cause and effect. One change can just ripple through a whole ecosystem.
Exactly. Yeah, it's all connected.
Okay, so let's tackle question 3C,
Right.
which dealt with osmosis, specifically the direction that water moves.
Osmosis, yeah.
Yeah, a lot of students mess this up.
It's a tricky one.
Yeah.
The key principle here is that water always travels from an area of high water concentration to an area of low water concentration.
Okay.
So it's trying to even things out.
Okay, like it wants to be in equilibrium.
Exactly. It wants to reach a balance.
Okay.
So if you put a red blood cell in distilled water,
Okay.
the distilled water has a much higher water concentration than the inside of the red blood cell.
Right.
So water rushes into the cell,
Okay.
making it swell up, and sometimes it can even burst.
Oh, wow.
But if you put that same cell in a concentrated salt solution, the opposite happens.
Okay, why is that?
Well, because the salt solution is more concentrated.
Okay.
The water moves out of the cell to try and balance things out.
Okay.
And that causes the cell to shrink.
So it all comes down to understanding that concentration gradient.
Exactly. Yeah, it's all about the concentration.
Okay, next up is a topic that seems to always trip people up.
Oh, really? What's that?
The nervous system.
The nervous system.
So surprisingly, a lot of students couldn't identify the withdrawal reflex for question 4.
Oh, yeah, the withdrawal reflex.
Wow.
Well, it goes to show that you need to be familiar with key terms and definitions, even if you understand the concept itself.
It's like trying to explain something in a foreign language without knowing the right words.
Exactly. Yeah, you need the vocabulary.
Okay, so let's break down how nerve impulses work.
Okay.
So we're talking about neurotransmitters diffusing across synapses.
Right, those tiny gaps between neurons.
Exactly. And the direction that a nerve impulse travels is super important.
Absolutely. It only goes one way.
Yeah.
And the examiners even included a calculation about impulse speed and neuron length.
Oh, really?
Yeah, so it just shows how math sneaks into biology sometimes.
That's all connected.
It is.
All right, shifting gears to question 6, which focused on genetics.
Okay, genetics.
Some students had a really hard time with those genetic diagrams,
Oh, yeah, I can see why.
especially when it came to representing alleles correctly.
Right, yeah. You got to remember, capital letters are for dominant alleles,
Okay.
and lowercase letters are for recessive alleles.
So it's like the secret code of genetics.
Exactly, it's like a code.
And there was this interesting observation about the ratio of axial to terminal flower plants in one of the crosses.
Oh, really? What was that?
The observed ratio didn't quite match the expected 3-to-1 ratio.
That's interesting.
Yeah.
Why do you think that is?
Well, the examiners said that fertilization is a random event and environmental factors can influence things like germination rates.
Ah, so biology isn't always as predictable as we'd like.
I guess real life is always a bit messier than those neat Punnett squares.
Exactly. Yeah, nature has a way of throwing curveballs.
Yeah, for sure.
Yeah, it can be tough. And questions 6C and 7, those ones about experimental design really tripped some students up.
Oh, yeah. Designing a solid experiment can be a challenge, for sure.
Yeah, it's all about those variables.
Right.
You need to be crystal clear about your independent variable,
Okay.
the thing you're changing,
Right.
the dependent variable, the thing that you're measuring,
Okay.
and the control variables, all the things you keep the same to make sure your experiment is fair.
To make sure it's a valid experiment.
Exactly.
Okay.
So the report uses the example of investigating the effect of temperature on lipid digestion. So the temperature would be your independent variable.
Makes sense.
The rate of digestion would be your dependent variable.
Okay.
And things like the amount of lipase, the amount of lipid and the pH, those would be your control variables.
Gotcha. So it's really important to keep all those other factors consistent.
Exactly. And the examiners really stressed being precise about how you control those variables.
Oh, really?
Yeah, it's not enough to just say you'll keep them the same.
Okay.
You have to explain exactly how you'll do that.
So, for example, using the same volume of lipase solution each time.
Exactly. Or using a buffer to maintain a constant pH.
Right. So it's all those little details that really make a difference.
They really do.
Okay, let's move on to anaerobic respiration
Okay.
and those massive industrial fermenters.
Yeah, those huge tanks.
I know, they're used to produce so many things.
I know, it's amazing.
Question 8 showed that while students generally understood the concept, they weren't always using the most precise language.
Yeah, the examiners pointed out the importance of using specific terminology,
Okay, like what?
like optimum temperature,
Okay.
or preventing overheating rather than just saying temperature control.
Oh, right. So it's not just about knowing the general idea,
Mhm.
but demonstrating a deeper understanding of the process,
Exactly.
using the right term.
Yeah, and it all ties into how those industrial fermenters work in the real world,
Yeah.
To get the most product out of those microorganisms, you have to really carefully control things like temperature, pH, and sterility.
It's all about creating the ideal conditions for those little workers.
Exactly.
It's amazing how much we rely on microorganisms for so many things.
Isn't it incredible?
Okay, ready for the final challenge?
Yeah, bring it on.
Question 9, which focused on genetically modified bacteria and insulin production.
Okay.
That was a real head scratcher for a lot of students.
Yeah, it's a complex process for sure.
Yeah, so let's break it down,
Okay.
step by step.
Sounds good.
First, you need to use restriction enzymes.
Right, those are like molecular scissors. They cut out the desired gene.
So in this case, the gene for human insulin.
Exactly. You're snipping out the gene you want from its original DNA source.
Okay, so we've got our gene,
Now you need a vector,
a vector.
which is like a delivery vehicle to carry that gene into the bacteria.
Oh, okay.
Plasmids, those little circular pieces of DNA found in bacteria, they're often used as vectors.
So it's like a tiny taxi service for genes?
Exactly, a taxi service for genes. I love that.
And to get that gene into the plasmid, we need another enzyme called ligase.
Right? It's like molecular glue.
It sticks everything together.
Exactly, you got it.
So once the gene is safely tucked into the plasmid, you introduce it into the bacteria,
Right.
and then they can start producing human insulin.
It's amazing, isn't it?
It really is. But why do people with diabetes have to inject insulin instead of just taking it as a pill?
That's a great question.
Yeah.
It's because insulin is a protein.
Yeah.
So if you swallow it,
Uh-huh.
it gets broken down in your digestive system, just like the protein in your food.
So it needs to get directly into the bloodstream.
Exactly. Injecting it bypasses the digestive system. That makes sure that the insulin reaches its target cells and can do its job regulating blood glucose levels.
Wow. It has been quite a journey dissecting those GCSE biology exam blunders.
It really has. But remember, mistakes are just stepping stones to a better understanding.
Yeah.